Hello, my name is Vivian Feig, and it's my pleasure today to talk about my graduate research on conductive hydrogels for next-generation bioelectronic interfaces. Throughout my PhD, I've been interested in the question of how to bridge the gap between electronics and the biological world. While there are a lot of exciting opportunities that exist at this intersection, our ability to fully leverage these possibilities is limited by the fact that conventional electronic materials have mechanical properties that are vastly different from those of biological systems. First, electronic materials are typically very stiff, whereas biological tissues like brain tissue can be as soft and squishy as jello. Second, while electronic materials typically cannot be stretched significantly before they break, much of the body comprises highly dynamic and moving and stretching surfaces. And finally, there's a mismatch in the dimensionality of the interfaces formed with these kind of materials, with conductive materials typically forming planar or two-dimensional interfaces, whereas cells in the body interface with each other in all three dimensions. So in today's talk, I'll focus on these three properties of stiffness, stretchability, and dimensionality, and I'll discuss why they're important for us to think about when we're developing next-generation biointerfacing electronics. I'll then share how I've approached bridging these gaps through my work developing new materials in Professor Jinan Bao's group at Stanford. In particular, I'll show that by just manipulating and processing a common commercially available polymer system, we can create conductive and patternable materials with tissue-level stiffnesses, highly stretchable conductors with orthogonally tunable mechanical properties, and injectable conductors that can be interfaced with cells in all three dimensions. So first, I'll talk about stiffness. Now, conventional conductive materials like metals can be hundreds of millions of times more stiff than soft biological tissues. And this mismatch in stiffness makes it very challenging to have long-term stable bioelectronic interfacing. For example, if you implant a stiff electrode into the brain, that mismatch causes an inflammatory response that leads to glial scar encapsulation of your device and ultimately to loss of functionality. This mismatch can also locally impact cells in a negative way. For instance, cellular biologists have shown that mechanotransduction mechanisms allow cells to sense and respond to the stiffness of their surroundings. And so you can imagine that if you interface cells with materials that are millions of times more stiff than their native tissue environment, that will naturally impact the ways that the cells differentiate and grow. Now, one way to resolve this mismatch is to use a type of material called hydrogels, which are materials that comprise hydrophilic polymer chains and hold a majority of their weight in water. Because water acts like a plasticizer for the polymer chains, hydrogels can be as soft and flexible as biological tissues, and indeed are already ubiquitous in biointerfacing products and technologies. To make hydrogels electrically conductive, we rationalized that we would need to form macroscopically connected networks that are capable of electrical charge transport while still retaining that high water content we need for these very low modulus values. And the way we achieve that is by working with a conjugated polymer system called P.PSS, which is a two-component system where P. can conduct electrical charges along its conjugated backbone, and PSS acts as both as a dopant for P. and as a way to disperse it in water. P.PSS is sold commercially as a dilute aqueous dispersion, and in water it takes the form of these dilute microgel particles that repel each other due to electrostatic repulsions from sulfonate groups that are negatively charged on the PSS. And that allows it to behave in solution like a viscous liquid. Now, it's previously been shown that if you mix these P.PSS solutions with ionic additives, including those found in phosphate buffered saline solution, those ions screen the electrostatic repulsions between these microgels, causing them to aggregate into a solid-like hydrogel that has a storage modulus around 100 Pascal and tan delta values less than 1. However, despite forming a percolating polymer network from a rheological standpoint, hydrogels made from these ions in PBS are actually very poor electrical conductors, with conductivity values around 10 to the minus 5 Siemens per meter. And with that observation, we were interested to see if we could manipulate the morphology of these gels to enhance their conductivity while still maintaining that very low storage modulus. Based on our knowledge of processing P.PSS into thin films, we hypothesized that forming P.PSS hydrogels from rationally selected ionic additives would also enable us to enhance their electrical conductivity as well. In particular, the Bao group has previously showed that certain ionic liquid additives can significantly boost thin film conductivity when they're blended with P.PSS and spin-coated into films. What these ionic species do is disrupt that electrostatic interaction between P. and PSS, enabling them to phase separate and form these highly conductive fiber-like networks where the fibers comprise crystalline P.rich regions where the P. is interacting with each other through pi-pi stacking interactions. And so based on these insights, we decided to synthesize hydrogels using one of these conductivity enhancing ionic liquids. And we compared it to gels that were made from PBS salts, which don't have a conductivity boosting effect on P.PSS thin films. Our results show that in both cases, we can make these ultra soft hydrogels with comparable mechanical properties, including storage modulus values around 100 Pascal and tan delta values much less than 1. However, the ionic liquid form gel now had a conductivity that's 5 orders of magnitude higher than the gel made from PBS, despite having these very similar tissue-like stiffnesses. Thus, in contrast to the poorly conductive hydrogels, where the network is forming from aggregation between dilute microgel particles, I argue that these kind of ionic species can transform P.PSS hydrogels into having an electrically conductive morphology comprising an extended pi-pi stacking-based conductive network. Beyond being able to manipulate the electrical properties of our hydrogels, we also found that different ionic species result in dramatically different kinetics of P.PSS gelation, which we can take advantage of to pattern our conductive materials in a couple of different ways. For one, we found that the same ionic liquid results in slow gelation of P.PSS with a 13-minute working window after mixing where the precursors remain liquid-like and flowable. This enables us to pour the gel precursors into molds, allowing us to obtain freestanding gels in a variety of different shapes, which means that we can directly mold these conductive gels into things like electrode patches for physiological monitoring purposes. On the other hand, we found that gelation in the presence of metal cations happens nearly instantaneously. And we use this fact to devise a new electrogelation patterning method where we take a sacrificial metal layer and oxidize it in the presence of P.PSS electrolyte. This oxidation generates metal cations that locally cause the P.PSS to form a gel. The advantage of this kind of method is that now we can use very common metal thin film patterning techniques like thermal evaporation through a shadow mask to first create that sacrificial pattern. And then after oxidation, the fast gelation of P.PSS in the presence of those metal cations enables us to reproduce that original sacrificial pattern in the gel with high fidelity. We can therefore use this method to pattern high-resolution features on substrates with feature sizes down to 100 microns, and to form conformal conductive coatings onto 3-dimensional structures, including this coiled wire shown here. And so overall, what these examples illustrate is that rationally selected ionic additives enable us to design and pattern new electrical conductors that have ultra-low tissue-like stiffnesses. Beyond stiffness, the next property that's important for biointerfacing is stretchability. And the reason it's important is because many of the parts of the body that you'd be interested in transmitting signals across regularly experience large strains, including the surfaces of the heart and skin. Even your brain, which you don't typically think about as being in motion, actually also experiences quite a bit of displacement just from respiratory activity. To make electronics stretchable, researchers have previously used geometric techniques like serpentine or mesh-like structures that can accommodate strain, but these kind of strategies inherently limit the density and complexity of devices that can be realized. And so because of that, there's been a lot of interest in intrinsically stretchable conductive materials, although combining stretchability with tissue-level stiffnesses in the kilopascal range remains quite a bit of a challenge. So to address this challenge, what people have done in the past is taken known stretchable hydrogels that are electrically not conductive and added some conductive fillers into them as an additive. But in these cases, the lack of a well-defined conductive network resulted in very poor conductivity values, less than 1 Siemens per meter. So we therefore asked ourselves the question, how can we leverage these known hydrogels that are soft and stretchable and combine it with what we know about making soft conductors to make gels that are simultaneously soft, stretchable, and conductive? To solve this problem, we decided to interpenetrate our soft P.PSS hydrogels with a second polymer network that's known to form stretchable gels. And specifically, we backfilled P.PSS gels with the monomeric precursors for a covalently cross-linked polyacrylic acid network, which we then polymerized to form a material that we call CIPN, or conductive interpenetrating network gels. These CIPN gels were highly stretchable to over 100% strain, soft and conductive, with values above 10 Siemens per meter, which is much higher than those previous approaches where the conductive component is used as a filler and not actually made into a network. Beyond that, we could also tune the modulus over 3 orders of magnitude by just changing the ratio of acrylic acid monomers. And because we've separated the conductive network from the stretchable one, we're able to do this while maintaining our high conductivity values within the same order of magnitude, despite these very large changes in mechanical properties. Therefore, not only does this approach enable us to make soft and stretchable conductors, but it also gives us a lever to be able to orthogonally tune properties via that secondary polymer network, which we've decoupled from the network that dictates the conductivity. So now that we've talked about stiffness and stretchability, the final property I like to discuss is dimensionality. And the argument I'd like to make today is that there is a real need for biointerfacing conductors that allow for 3D interactions with biological systems like cells, because that not only increases the density of interactions that you can have, but it's also just a much better representation of how these cells actually interact with each other and with their environment, which is why tissue engineers more generally already largely use 3D scaffold materials in their work. Despite this, conventional conductive interfaces remain two-dimensional. And I think a major reason for that is because, in general, existing strategies for forming 3D cell niches rely on cross-linking interactions that are incompatible with the macroscopically connected conjugated networks that we need for good electrical conductivity. These strategies include having cytocompatible cross-linking mechanisms where a scaffold forms directly around the cells, or using gels that can be mixed with cells because they have supramolecular cross-links like guest-host interactions that allow them to be repeatedly injected. So when researchers have used these kind of strategies in the past to form conductive 3D scaffolds, the conductive component is inevitably relegated to the role of a filler that does not actually form into a macroscopically connected network. And that explains why the reported conductivities have been very low, less than 1 Siemens per meter in the best case. So we hypothesized that we can make repeatedly injectable scaffolds that were also macroscopically conductive if we use granular hydrogels. Granular gels are made from jammed micron-sized gel particles, where at this micron size scale, frictional forces between the particles cause the entire system to behave elastically despite not being chemically cross-linked together. And so you can get this combination of elastic behavior, but also deformability. This is analogous to how if you compact rice grains into a container, you can actually lift them up all together using a single chopstick. And so our hypothesis was that if we could form granular gels using conductive hydrogels as the building blocks, then the junctions between these jam particles would not only introduce dynamic elasticity to the bulk material, but they would also introduce bulk conductivity because each of those building blocks is also individually conductive. As a proof of concept, we created granular conductive gels through a very simple approach where we start with a conductive P.PSS hydrogel and mechanically fragment it into heterogeneous micron-sized particles. The fragmented gel exhibited rheological properties that are consistent with what we expect for a granular material, including very good shear thinning behavior and the ability to alternate between solid and liquid-like behavior at high and low strains, meaning that the gel can be injected through a syringe and then rapidly recover its solid-like behavior. Remarkably, in spite of our fragmentation process, our gels retain a very high conductivity of 10.8 Siemens per meter, which suggests that a large portion of the conductive crosslinks in the material are retained in these fragmented micron-sized particles. Moreover, if we compare this value to previously reported conductive 3D scaffolds, we can now clearly see the advantage of our design approach. Since the jammed microstructure provides a macroscopic macroscopic network of conductive building blocks, we can achieve conductivity values that are now 2 orders of magnitude higher than in these previously reported examples. To further understand what's happening, we used electrically conductive parallel plates to monitor the electrical resistance of the gels while we measured their rheological properties at high and low strains. And what we found is that when the material is behaving like a solid at low strains, the resistance is also low, whereas when it's behaving more like a liquid and sheared into these high strain values, the resistance jumps up. And so this direct correlation between the electrical and mechanical properties further validates our hypothesis that it's the jamming-induced elasticity in these materials that's also responsible for that highly conductive network. It's also why we can inject our granular gel through a syringe and have it immediately be conductive enough to complete a circuit and light up an LED. And so using our gel, we've been working with collaborators at the Stanford Medical School to test the viability of encapsulating neural progenitor cells within the scaffold by mixing it with our gel. And what we found is that there's no significant difference in viability compared to just cells alone after 5 days. And so moving forward, we're really excited to continue working together with them to study how these cell-laden conductive 3D scaffolds might be used for regenerative medicines, including repair of neural activity after stroke. So overall, through my PhD, I developed an understanding of how to design and pattern new hydrogel materials that are not only ultra soft, but also electrically conductive. Building on these hydrogels, I showed that we can use interpenetrating polymer networks to make stretchable conductors that also have orthogonally tunable mechanical properties. And finally, I showed that we can use these conductive gels as building blocks to make granular gels that can be injected and interface with cells in 3 dimensions. I'd like to acknowledge my advisor, Professor Jianan Bao, my mentors, Dr. Helen Tran and Dr. Mina Lee, as well as my longtime mentee, Kathy Liu. I'd also like to thank my collaborators in the Stanford Med School, Professor Paul George and Dr. Shruti Santhanam, as well as all of my other co-authors and my funding sources, the NDSCG Graduate Fellowship and Stanford BioX. Thank you all for your attention.